Hi, this is Miss Litton, and this is 7th period, wonderful AP biologist, and we are reviewing Chapter 11, Mendelian Genetics, Mendelian Patterns of Inheritance, and Exceptions. Say hi. Hi. I hope you're having a lovely Tuesday. So let's talk a little bit about Mendel. Just historically, before him, traits were thought to be about blending. You know, and just blended it together of the parents. Didn't really understand about chromosomes, but it was Mendel who came up with the idea. Oh, you can't see because this is frozen. Okay, this makes sense to me. About the particulate theory of inheritance, that there were individual units. You and I call these alleles, right? We call those alleles. But he didn't understand about homologous pairs. He did most of his work on P's. Great at math, great at recording data. He would cross true breeding plants for generation after generation of tall. So he knew for sure they always made tall babies. Generation after generation of short, they always made short babies, and then he would cross them. Then when he did something like that, true breeding tall with a true breeding short, in that first generation in the F1, what trait seemed to be gone? Short. Okay? But then when he took the F1s and crossed them together to form F2s, what trait showed up? Short. Okay? And it's the typical geno and phenotypic ratio. Would you know what that is? Yes. That's called a monohybrid cross. A monohybrid cross, and you would want to be able to recognize, okay, mono meaning one, right? A monohybrid cross. Okay, so the genotype ratio is what? Genotype ratio. What do you hear in genotype? Genes. Let's talk about those genes. So tell me what it is. Two what?
Big T, small t.
Okay.
Little t.
Yes. In a typical monohybrid cross, it's not incomplete dominance. It's, your typical monohybrid cross. Your genotype is always going to be 1 to 2 to 1. Also, in a typical monohybrid cross, your phenotype will always be what? 3 to 1. 3 tall to what? 1 short. Okay? 1 to 2 to 1 and 3 to 1 is a typical monohybrid cross, and that's what you would predict if there was no other, things coming into play. And we'll talk about those exceptions as well. Okay, ask your bio buddy next to you. Is that lemon squeezy to you? Easy peasy, lemon squeezy. Now, we learned about 3 laws or principles. Okay, what law is this? Law of dominance. Okay, that law, is saying that the tall allele, if you're pairing a tall and a short allele together in a zygote, the tall allele would be dominant over the short allele. So when you have a hybrid, they would appear to be tall. Are there exceptions to that rule? Are there exceptions to the law of dominance?
Yes.
Yes. Give me an example. Let's talk about just that right there. From what we know in class, give me an exception. Yes? Codominance. Okay? What is a real-life example of codominance?
Wavy hair is codominance.
Oh, go again. I'm looking for something else, but you're going to come back to that one. It's still good. Yes?
Blood type.
What? Blood type? Is that what you're telling me? Yes. Give me a real example in blood type.
When you have B and O alleles.
No. Give me something else. Yes?
A and B?
A, B. Because both the A allele and the B allele are dominant, right? When you have type A blood, you can have big IA, or you could have big IA, little I. That's still dominance, right? That's still dominance. Same thing for B. Where you see where your you're not adhering to the law of dominance right here. The exception is codominance. Big I A, big I B. Both the A and the B allele are expressed, and those are expressed as antigens on the red blood cell. Okay, another exception. Another exception. You were referring to it, you just had the wrong name. Yes? Incomplete dominance. Okay, what is the real life example of incomplete dominance that we looked at? Yes?
Wavy hair.
Yes, wavy hair, okay? So wavy hair, remember, is H1, H2, whereas straight hair is H1, H1 for the alleles, and curly hair is H2, H2. But wavy, incomplete dominance, the best, the way to explain that is it would look like blending. A red flower crossed with a white flower, and you get a pink flower. That's incomplete dominance. So that is another exception to the law of dominance. Lemon squeezy? Yes?
Is incomplete penetrance?
Okay, so we can talk about penetrance and expressivity. I wouldn't really, I mean, you could put that in there. When you talk about penetrance, you're saying how often is that trait expressed, right? So there could be exceptions where you had the allele and it's a dominant allele, but it doesn't get expressed. So yes, I would use that. These would be more classic examples, but I can see that. And then expressivity is how it is expressed. Okay, I'm looking for another exception. It has to do with boys, biological boys, and biological girls. Yes?
When you have a recessive, if you're a guy and you have a recessive X trait, even though you only have one recessive, it'll get expressed as being X.
That's right. You have a single X. So when you talk about biological males, okay, they could have X little b y. Normally, to express that recessive trait, you would need 2 copies of that allele to express that recessive trait, because if you just get one dominant allele, you wouldn't express it. But gentlemen, since they only have one shot at their X, if they get it, they get it. Okay? These are all great examples of exceptions to the law of dominance. Okay? But any questions on that part? Ask your bio buddy if they're okay. Any shadowlands? Do you need the flashlight of wisdom to be shown upon you? All right. Okay, let's talk about another law. What is another law that we know about? Law of what? Law of segregation. So when you talk about the law of segregation, in the formation of gametes, the homologous pairs are separated from each other. The alleles are separated from each other. Okay? That is a true statement. Are there exceptions to the law of segregation? In any normal conditions? No. Okay? What would we call that failure to separate? Non-disjunction. And that is not a normal process, right? That is something that's not working well. When we talk about the most common form of non-disjunction in the autosomes, what would that be? Trisomy 21. But that is not as it was intended. We still love them, but it was not as it was intended, right? So the law of segregation, there aren't really exceptions to that. Not in any of the normal processes. Because if you're going to do meiosis correctly, in anaphase 1, you will separate the homologous pairs, and in anaphase 2, you will separate the sister chromatids. Okay? Questions or concerns? Okay, let's talk about another one. There's a law of segregation. What was the third one? Law of independent assortment. So that's saying if you have something like this, that the gametes you could get is you could get a big A with big B, a big A with little b, a little a, big B, little a, little b. That you could have any combination of alleles in the gametes. Any combination of alleles in the gametes. But that is only true if the particular pair of alleles you're looking at are on 2 different pairs of chromosomes, right? Because the exception to that rule would be what? On the same chromosome. And do you know what we call that? What kind of genes those are called? Starts with an L. Linked genes. Yeah. So your exception to the law of independent assortment is linked genes. linked genes. Okay? Because if you have a homologous pair, okay, and if on here you have big A, right, these are sister chromatids, so they're going to be the same, right? So when you have linked genes like that, big A and big B are going to travel together, and little a and little b are going to travel together, right? So you're probably either going to get, mixing it up, you're probably either going to get, half of them to be completely dominant and half of them to be completely recessive, right? But what could then break up linked genes? Crossing over. So crossing over, okay, so independent assortment says I should be able to get big A with big B, big A with little b, but linked gene says no. Then what says no to linked genes is crossing over. That would give you that chance to get a big A with a big B or a big A with a little b. So these are Mendel's 3 laws. Law of dominance, we can look to see where there would be exceptions to that. Law of independent assortment, we can see where there are exceptions to that. And then we can see we don't see any normal exceptions to the law of segregation. Yes?
Would linked genes be the cause for non-disjunction?
No. Linked genes are just linked genes. So they wouldn't cause you to fail to separate. What would be more than likely the failure to separate would be the age of the egg when meiosis II took place. That would be, and the size of the chromosome. If it's really small and there's a lot of surface area where it's sharing, it's kind of hard to pull it apart if it's a really old egg. Okay, questions on that part? Okay, so then that was the first part. Then, let's see. Then we went into what are the reasons for a test cross? What are the reasons for a test cross? Yes?
To check if they're homologous, if they're not homozygous dominant, or if they're heterozygous.
Right. And so your reason is you don't know the genotype of a dominant phenotype, right? When you don't know the genotype of a dominant phenotype, that's when you have to do a test cross, right? You don't need to test somebody who has blue eyes. You know they have blue eyes. They're a little b, little b. Because in order to express that trait, they have to be homozygous recessive. But somebody who has brown eyes, we don't know if they're brown or they're brown what? Blue. So who do you always test? Like what's a standard? Hey, I need to do a test cross. What should I use? Take eye color out of it. Just use your descriptors. I need to do a test cross. What should I use? Homozygous recessive every time. Homozygous recessive. Now, give me what would you predict? What kind of information in your results would help you solve this problem? Yes?
Well, if all of your offspring have the dominant trait, then you know that most likely it is the homozygous dominant.
Perfect. And if
some of them have the recessive phenotype, then they for sure have to have the dominant and recessive.
What would you call that?
Heterozygous
Boom.
genotype.
Okay? Questions on that? Questions or concerns on that? All right. So then let's move on from that. So then we talked about, remember this family? Score 1 for the Lytton on that one. Good job. How did they inherit that trait? I don't know. Let's go ask their real parents. Okay, now. They're their real parents. Okay. Now, when you do your crosses, you would want to look at your data. Like if they give you some data and they say, hey, this is what I think the cross is. For instance, I think this is incomplete inheritance, this particular trait about hair type. So this, let's just say rats, okay? This family of rats, they had, let me draw some rats. I don't even know how to draw a rat, okay? This family of rat, they had wavy hair. This family of rats, okay?
No, I do see it now.
Do you see my rat?
I do see it now.
Okay. Here are two wavy rats. And these wavy rats got married. Okay? They had many different offspring. Be with me. I don't know how to do this exactly right. When I counted up all their rat babies, 20 had straight hair. They were prolific. 30 had curly hair. And 65 had wavy hair.
Oh my god. That's quite a bit.
Now, is there anything up with these rats, or is this what we would predict? So I want you to think right now, how would you go about solving this problem? Should I have concerns about these rats, or are my rats, besides how prolific they are my rats okay?
They wouldn't have
I'm going to pause this just for a minute. All right. So I predict that this is incomplete dominance. Do you agree with that, Donna? Okay, so this right here, we could say that these are observed, right? Are you okay? These are observed. So what would we expect for each of these? Well, if I cross two wavy-haired rats, are you with me? Yeah? Okay, you with me? So I would expect 25% to have straight hair. Yes? I would expect 50% to have wavy hair and 25% to have curly. Yes? So how many rat babies are we talking about here? 115? Okay. So what's 25% of 115?
Around 29.
29?
It's 28.75.
Okay, 28.75. So then this must mean curly should be 28.75. Do you agree?
Yeah.
And then wavy has to be 50.
6.5.
Are you all right? Do you know what I did? Do you need me to repeat what I did? Okay, listen up, and if you know, just listen. If I cross two wavy-haired rats, H1H2 with H1H2, those are wavy, right? H1H1 would be straight, H2H2 is curly, right? So I do the Punnett square. When I predict 1 out of my 4, or 25%, would turn out H1H1, which is straight. 25% would turn out H2H2, which is curly. And 50% would turn out to look just like their parents, wavy. So then I said, how many rats do I have? These prolific rats made 115 rat babies. I would expect half of them to look just like the parents, 50%, right? I would expect 50% to turn out wavy. I would expect 25% to be curly and 25% to have straight. So I took 0.25 times 115 and came out with that many. So this is based on how many offspring they had. I would expect almost 29 of them to be straight, almost 29 of them to be curly hair, and I'd expect 57, 58 of them to have wavy hair. Yes?
How did you do that cross to figure out that you would...
Oh, it's because we learned in class that wavy hair was incomplete dominance. And I'm telling you, these are two wavy-haired rats, so for them to be wavy, they'd have to be H1, H2. And those two wavy rats are having sex together. Sorry, that sounded really weird. Okay. And so when you have two wavy ratted hair sex going on with rats, then that's how you, just like you did, remember when you predicted the monohybrid cross with me and we did the genotype ratio and the phenotype ratio? The thing about incomplete dominance is the genotype and phenotype ratios are the same, right? Because the heterozygous expresses its own phenotype. How are you doing with that? Okay? Yes?
So even though they're rats and you can't really divide them, you'd still put the decimals?
Yeah, and you could round if you wanted to, probably good, but just decimal, that's fine. Okay, so then all I did is I pulled out my handy dandy chi-squared, which I know is the sum of the observed minus the expected squared over what I expected, yes? Okay, so then I just had to take observed 20 straight-haired rat babies minus expected 28.75 rat babies, and what is that? 8 something? Yeah, 8.75 squared over what did I expect? 28.75, right? You know what I'm doing now? Okay, and then I had to take 28.75 minus 30 rat babies, and what did I get?
2.25.
Really?
No,
1.25 squared over 28.75, and then for the third one I had to do 65 minus 57.5. What is that? Squared over what I expected, 57.5. I do each of those. I add them up. What did you get?
Around 3.7.
Around what?
3.7.
Really? I thought I got more than that. Really? Is it 3.7? Can I get some verification? Because I didn't write it down. I can't remember what I got. Is that right? We got verification? Now, if we looked on our chi-squared, let's go find a chi-squared chart. I know I have one somewhere. Here I do. How many degrees of freedom did we have? 2. Because how many phenotypes did we have? 3. Take away 1 leaves us 2. What was our number? 3.7. Are we okay? Yay! Our rat babies are fine. Okay? So, it's a really good problem for you, right? Because what are all the things I could test you by asking you that? First of all, your understanding of incomplete dominance, your ability to understand between genome phenotype ratios, your ability to analyze your data, right? And to tell me whether or not that was a significant difference or not. And it turned out to fall into the insignificant because it would have, somewhere between 10 and 20% of the time, I could have had those exact rat babies. And so that, it looks like that kind of inheritance is incomplete dominant. So that worked out. Yes?
So what's the
You want me to go back to this?
No, it's okay. What's the left hand column? Like when you, how many possibilities there are? Like 1, 2, 3.
This one?
Yes.
Okay, that's however many different phenotypes you
So
have.
in this one there'd be 2 instead.
Well, we had 3 phenotypes,
Yeah.
curly, straight, and wavy.
Mhm.
So since we have 3 options, we take away 1, and so we're down to 2.
2 and
Let's remember when we did the P cross and I showed you the shortcut for the P's? Did I remember to push play? I did. Okay. Do you remember when we did the P's? We said you could be round and yellow, round and green, wrinkled and yellow, wrinkled and green. So how many phenotypes did we have? 4. So for that one, how many degrees of freedom would we have? 3. If I was doing tall versus short, right, how many options do we have? 2. And so how many degrees of freedom would I have? 1. Okay? Ask your bio buddy, how are you on that? Do you understand that part? That was a great problem I just came up with. I just couldn't say it. I was super impressed with myself on that one. Okay.
No, how many options do you have to basically?
Okay. Now, let's keep going. Where do I want to be? Okay, so let's talk about some other things that we learned about. Where am I? Come here. Okay, good. All right. Then we started looking at pedigrees. And I tried to review that a little bit with the rats. So the females are circles, the males are square. Whatever you're studying is colored in. When you're faced with a pedigree, they may not tell you how the form of inheritance is. It could be autosomal dominant, it could be autosomal recessive, it could be sex-linked recessive, it could be sex-linked dominant. I won't give you any of those. Those are the three options you'll always have with me. If I say, hey, how do you think this could be inherited? I'd expect you to be able to analyze that pedigree and be able to tell me, I think that is an autosomal dominant trait. I think it's autosomal recessive. I think it's sex-linked. Let's think about key things we would look for each of those. Like, how would I identify that? Okay, so I have auto, dom, auto, recess, and X-linked. Okay, what are some things you would look for in each of those that go, hey, I think this is this? What are some things? Yes.
For autosomal dominant, the parents would be shown like shaded in because it's.
Okay, think of a way you can tell me that. Yeah.
For autosomal dominant, two affected parents having an unaffected child.
Okay, you could have two affected parents have an child. Good. Okay, that is possibility. Could that happen with autosomal recessive? No. So if I go like this, no. Couldn't happen with autosomal recessive. Yes. You can give it in any one. I don't care.
Auto-recessive would be if two unaffected parents have a affected child.
Could that happen in autosomal? Two, what do you say it again?
Two non-affected.
Two unaffected, right?
Having unaffected.
Could have, yes, could have affected child. I agree with that. Could that happen in autosomal dominant? No. Good. Yes. Yes.
For X-linked males who change genetics.
Is that a true statement?
Well, if it's recessive.
It's recessive.
But
Males can only get it if it's recessive? Seriously?
Males
Come on
only
now.
get it with one, but females only two, so it's more predominant in males.
That's what you could tell me. More likely in males. What else? You could tell me some more things here. I know you can. Yes,
One affected for autosomal
gentlemen.
dominant.
I couldn't hear. What?
For autosomal dominance, you could write one affected parent and one affected child.
I don't know what you.
One affected parent and one affected child.
Okay, but I could have that for dominant too, right? Okay, so let's think of things that set them apart. What don't you see in autosomal dominant? It doesn't generations. Doesn't skip generations. Does not skip. Can this one skip? Yes. This one can skip. Are either one of these more likely in males? Autosomal dominant, autosomal recessive? No. You would not see this any more likely in males in either one of those. What could you tell me about a daughter of a if child inherits disease? What could you tell me about her parents? Yes.
If it's a girl, then her dad has to have it too.
Yeah. Sorry. If girl child. Yeah. Dad must have it. Because for a girl to have that disease, she has to get two bad X's. One of those bad X's coming from dad, so he must have the disease. Does her mom have to have it? No. She could just be a what? Carrier.
Her maternal grandpa has to have it.
Her maternal does he?
Yeah.
No.
Because the mom has to have it.
Her mom could have had it.
Her mom, but if her mom had a recessive.
But her mom could have a recessive and give it to her too. To her grandma. To her, the mother.
Oh, because the mom could be had.
Yeah.
Okay.
Okay, so those are some things we could see. Could these types of things should guide you, right, in how you see it. Could X-linked, I gave you X-linked recessive, could it skip?
Yes.
Yes, it could skip too. It could skip generation. All right, so those are our pedigree problems. All right, and then we talked about blood. Blood had two things for us to learn. We could learn two things from blood. Do you remember the two things we could learn from blood? We already talked about one of them. Yeah.
Was codominance.
Codominance was one thing we could learn from blood, is we could talk about codominance. And the codominance was right here, right?
Yeah.
So we could learn that from blood. What else could we learn from blood? Multiple alleles. Yeah. It's right up here. Okay? Multiple alleles. Because before, we were used to just having big IA, right, and maybe little I. But now we could have big IA, and we could have what? Big IB, right? So this was an example of multiple alleles. This would be like if we had tall and short, right? Big T, little t for P's. But now we have options on our alleles. We have more than two options. That's why it's called multiple alleles, because we have big IA, we have big IB, and we have little I. How does that affect? It changes our possibilities for our genotypes, right? We have a lot of different genotypes now because in any one pair of chromosomes, right? Oh, sorry, they had a deletion. In any one pair right here, you could have 3 different options, right? We could have any combination in each of those spots, where before we only had 2 options, right? So that gives us a lot more genotypes to work with, as well as phenotypes. So when you have 2 crosses, and you end up with several different phenotypes out there, then you go, hey, that's probably got to be multiple alleles, right? Because normally you would just maybe get 2 options. All right, ask your bio buddy if they're okay on that, if they have some questions. All right, and then the part we didn't like, I'll hit sex linked tomorrow in class, so don't stress about that. But I just have a couple more because I'll hit sex linked hard in class. That sounds terrible. Okay, so we already talked about. We already talked about penetrance and expressivity, so I'm going to leave that alone. So the last thing I need to talk about here is polygenic traits. And the way we remember that is poly means many genes. So it's not like we just have I's. We have I's and combinations of T's and combinations of Q's and combinations of R's that give us our total trait that we're looking at. Polygenic. So, for instance, when we look at colors of skin, we have 3 genes we're looking at. Some A's, some B's, and some C's. And so if you're very, 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 very light, you're homozygous recessive for all of those. If you're very, 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 very dark, then you're homozygous dominant for all of those. You all right with that? Now, what could influence it, even though I have a trait to be, let's say, medium? How could I end up not being medium at all? Yes?
The sunlight.
Okay. All right, so that could affect my melanin. No, there is another kind of gene that means, yes, do you know what it is? All right, so then E, yes. Epistatic. Yeah, epistatic. Standing upon. So you could have a gene that even determines if you get pigment at all. It doesn't give a rip if you're light or super dark or medium or orange. Okay? It doesn't care because it's the one that determines whether you get any pigment or not. Right? So it's great if you have this beautiful auburn hair, but if you have the gene for baldness, it doesn't matter. You're not going to get that hair, right? Does that make sense? Got it? All right. Yes.
Is that like albino?
Yes, albino.
Are we getting over this on the class?
No, I'm going over it right now. Okay. All right, so we looked at that. And then we looked at nature versus nurture. So you could have what your genes say, but some of the genes are turned on and off by things in the environment. So the temperature could influence whether you are a male or female. So the way that gene operates is dependent on something in the environment. So in this case, the Himalayan bunny, the parts of him that gets really cold, like his feet that walks on the cold snow, his snout because he's breathing in that cold air, and his ears, which are up there in the coldness, they're going to be that darker color. And that was evidenced, guys, seriously driving me nuts. When they shave their back and they put a cold pack on there, normally that area is very warm by the body temperature, right? But when they shaved his back and put the ice on there, it grew back black. Okay? So that's an example when we look at nature versus nurture. And then I mentioned that the polygenic traits, those traits where you have A's and B's and C's and Q's and R's, those tend to be more influenced by the environment than straight up other genes. Okay? And that was your epistatic one. And then that brings us to the sex-linked one, and that's where I'm going to start tomorrow in class. And we'll review from those on and do some crosses with those in class tomorrow. And then we'll finish up reviewing transcription and translation and talking about that tomorrow. And then we'll be done with everything. And then on Friday, we will start chapter 13. And by the way, the day of your test, I'm glad I made it really super simple because I have to go to another conference on that day. I know, I'm like, super. So, yeah. All right, so that's one of the districts sending me. I didn't sign up to go. What? It's a quadra-win. So now it makes me want to make it even easier, right? Of course. All right. Make the choices. Yeah. Write your name. 50 points. Have a piece of toast.